In this video, we'll discuss about vitamin B12 Cobalamin. It contains corin ring. It contains corin ring with central cobalt. Another name vitamin B12. Active form of vitamin B12 is adenosyl cobalamin. Second one is methyl cobalamin. These two things are active form of vitamin B12. Adenosyl cobalamin, methyl cobalamin. It required intrinsic factors for the observation in the ileum. So it required intrinsic factor for the absorption in the ileum. The deficiency of this vitamin B12 leads to various neurological symptoms. The deficiency of vitamin B12 leads to various neurological symptoms. Neurological manifestation, it is known as neurological manifestation. It is known as extrinsic factor of Castile. Extrinsic factor of Castile. It catalyzes three reactions. Of these, two are very important. It catalyzing three reactions. Two are very important. First one, methyl malonyl coenzyme A mutase. Methyl malonyl coenzyme A mutase. Second one is the methionine synthetase. Methionine synthetase. And deficiency leads to the megaloblastic anemia. Deficiency leads to megaloblastic anemia and subacute combined degeneration of spinal cord, and deficiency leads to excretion of methyl malonate in the urine, that is known as methyl malonate uric acid. These are important aspect about vitamin B12. So the deficiency, megaloblastic anemia, subacute combined degeneration of spinal cord, and deficiency leads to the excretion of methyl malonate in the urine, that is known as methyl malonate acidic urea. These are the important aspect about that vitamin B12. Now coming to important point about vitamin B12. The first one is how it will digest, absorb and transport. So first important point is digestion, absorption and transport of vitamin B12. First, that salivary gland. Salivary gland secrete that important substance that is known as R-binder. Salivary gland secreted important substance, that name is called as R-binder or hepatocorrin. Hepatocorrin or cobalamin. Hepatocorrin or cobalamin. And the parental cell. Parental cell secrete intrinsic factor, and the two factors important for digestion and absorption. These two factors are important. What are the things? Salivary gland secrete important substance, name is called as R-binder or hepatocorrin or cobalamin. Second one is parental cell secrete intrinsic factors. These two factors are important for digestion and absorption of vitamin B12. When we take dietary vitamin B12 with action of that HCl, hydrochloric acid and pepsin, there is release of vitamin B12 in the stomach from the food. So when we take dietary vitamin B12 with the action of HCl and pepsin, there is release of B12 in the stomach from food. This B12 get bind with R binder in the stomach, transport up to the duodenum, where with the action of protease the complex broken down, and here the B12 get attached to that intrinsic factor which is released by the parental cell, gastric parental cell. Then it reaches that ileum. The ileum, there is a special receptor is known as Cobalamin. Receptor name is called as Cobalamin receptor. This receptor is present ileum cell on the intestinal site. When that intrinsic factor and B12 reaches this Cobalamin with the help of endocytosis. This receptor is present at ileum cell on the intestinal side. When the intrinsic factor B12 reaches that Cobalamin with the help of endocytosis, this complex get internalized. This complex get internalized to that ileum mucosa cell. From this, the cell get to the portal circulation. So what is the summary? R binder, it is secreted by salivary gland. Intrinsic factor secreted by this gastric parental cell. In that action of HCl and pepsin, there is release of vitamin B12 from food. It is getting attached with R binder in the stomach, transported up to that duodenum. In duodenum, it get to that intrinsic factor. When it reaches the ileum, there is Cobalamin receptor. In ileum, Cobalamin receptor, this intrinsic factor get attached to that Cobalamin receptor. With the help of endocytosis, it get internalized to that ileum mucosa cell. To that ileum mucosa cell, it get absorbed to that portal circulation, where it bind with that in portal circulation, it will bind with transcobalamin 2. Through this transcobalamin 2, it get reaches to the different tissue. So this is the digestion, absorption and transport of vitamin B12. So R binder secreted by salivary gland, intrinsic factor secreted by gastric parental cell, that action of HCl and pepsin. Whenever we are eating vitamin B12 food, it get absorbed from that R binder. In that stomach, transported to that duodenum. Then it will transported to the duodenum. In duodenum, it get to that intrinsic factor. When it reaches that ileum, there is Cobalamin. Cobalamin is the receptor. This intrinsic factor get attached to that Cobalamin receptor, and with the help of endocytosis to that ileum mucosa cell get absorbed to that portal circulation, where it bind with the transcobalamin 2. Through this transcobalamin 2, it get reached to the different tissue. So this is the digestion, absorption and transport of vitamin B12. Now coming to dietary sources, it is mainly produced by microorganisms in that intestinal flora of animals. So mainly produced by microorganism in the intestinal flora of animal. That's why it presented in the food of animal origin, that is meat, fish, etc. So it mainly produced by microorganism in that intestinal flora of animal. That's why it is presented in that animal food material. Next, RDA 1 microgram. Coming to that Coming to the biochemical functions of vitamin B12, vitamin B12 is responsible for catalyzed the two important reaction. The first important reaction is catalyzed by methionine synthetase. This is methyl transfer reaction. This one is the methyl transfer reaction in which homocysteine in this homocysteine is converted into methionine. In this reaction, this methyl THF tetrahydrofolate required. So this reaction required methyl tetrahydrofolate which get converted methyl tetrahydrofolate which get converted into tetrahydrofolate. Methyl tetrahydrofolate converted into tetrahydrofolate. For this vitamin B12 is required. So this is methyl transfer reaction, also known as homocysteine methyl transferase or methionine synthetase. It's B12 and tetrahydrofolate dependent reaction. The deficiency of this vitamin B12, there is increased level of homocysteine. This one leads to serious cardiovascular related diseases. And there is deficiency of vitamin B12. This THF get trapped as methyl THF because methyl THF reaction is irreversible reaction. It get black methyl THF and this is known as functional folate deficiency, that is known as folate trap in the deficiency of vitamin B12. Second important reaction, it catalyzed by methyl malonyl coenzyme A mutase. Methyl malonyl coenzyme A mutase. This methyl malonyl coenzyme A causes conversion of methyl malonyl coenzyme A to succinyl coenzyme A. And this succinyl coenzyme A enter to TCA cycle for complete oxidation and energy production, and also it can enter to heme synthesis. This succinyl coenzyme A enter TCA cycle for complete oxidation and energy production, and also enter TCA. This also enter for heme synthesis. Then prophenyl coenzyme A, which is produced by that oxidation of all chain that fatty acid, is converted into methyl malonyl coenzyme A by prophenyl coenzyme A. Prophenyl the coenzyme A is converted into methyl malonyl coenzyme A. This methyl malonyl coenzyme A is converted into succinyl coenzyme A. Vitamin B12 dependent and deficiency, this one is the reaction vitamin B12 dependent one. Deficiency of this vitamin B12, that methyl malonyl coenzyme A excreted in urine, that is known as methyl malonate acid urea. So deficiency of this vitamin B12, the methyl malonyl coenzyme A excreted in urine. So this condition name is called as methyl malonate acid urea. Coming to the deficiency, that manifestation, first deficiency causes of symptoms. First important causes of vitamin B12 deficiency, vegetarian diet. So nutritional diet, a person with strict vegetarian diet, there is deficiency of vitamin B12. Second one is malabsorption syndrome. Third one is increased demand. As in pregnancy, increased demand in vitamin B12. Fourth important causes pernicious anemia. In pernicious anemia, there is production of auto antibodies against that parental cell. That's why there is deficiency of intrinsic factor. And due to this deficiency of intrinsic factor, there is deficiency of vitamin B12. This is another cause of vitamin B12. So here second one is malabsorption, third one is increasing demand, mainly pregnancy period. Fourth important causes because of pernicious anemia, the production of autoantibodies against the parental cell. That's why there is deficiency of intrinsic factor. And due to this deficiency of this intrinsic factor, so what will happen, there is deficiency of vitamin B12 is the another causes of vitamin B12. Now coming to deficiency syndrome, first important symptom is megaloblastic anemia. It is due to that fact that vitamin B12 is related to folate, and due to the folate deficiency there is decreased DNA synthesis. Due to decreased DNA synthesis, cell arrested in S phase and continue to grow without cell division. That's why there is increasing in cytoplasm that leads to the macrocytic RBC, and number of immature RBC are there in the circulation. So first one is megaloblastic anemia due to that vitamin B12 related to folate. Due to folate deficiency, decreasing DNA synthesis, decreasing DNA synthesis, cell arrested in S phase, so continue to grow without cell division. That's why increasing in cytoplasm, so immature RBC in that circulation. Second important deficiency manifestation, that is subacute combined degeneration. This is due to defective myelin synthesis. First important cause of deficiency of myelination is methylmalonate coenzyme, which is generated due to deficiency of vitamin B12. It complete this malonyl coenzyme A inhibit fatty acid synthesis and inhibit synthesis of myelin. Last one is laboratory diagnosis. Serum sample taken, cobalamin, methylmalonic acid, homocysteine, bone marrow and peripheral smear. Thanks for watching.